speaker is Professor Graeme Hammer, who will talk to us about G2P modelling. Thank you, Graeme.
Thank you. And good morning. I want to talk about genome-to-phenome modelling and prediction, particularly in relation to enhancing rate of genetic gain. And I think Charlie has sort of introduced this topic beyond what I can do in 10 minutes, but I'll let you know what we've been doing around this field in the centre. There's a suite of collaborators there. The three names at the top are the three people in this section so I'll just try to introduce how we're going to sort of fit together. The second line are my ecophysiology friends, Eric and Andy, and then the software programming people, Greg, Jason, Joe, then the sorghum genetics and breeding crew at Hermitage, and then the group leaders, Mark and Christine, who connect both the breeding and genomic prediction, G2P work, and through to Christine's branching group where Nicole sits. Really, Mark, I suppose, is the sort of concept architect of this major project in the centre. And it comes back to this sort of diagram which looks at plant breeding, and quantitative phenotypic prediction is really critical inside plant breeding. And this is it's one of Mark's figures from that 2019 paper. I don't think I'll be able to use this thing. The whole point, though, is to have a reference population of genotypes, your RPG, which contributes to a multi-environment trial. So you've got a selection of genotypes. You test those in target population, a sample of your target population of environments, and you get some sort of phenotypic data there that enables you to analyse and potentially select and go around that cycle by crossing amongst those selected few to find the improved ones. The other loop in there is the genomic prediction loop, which is now really installed, where if you've got genotyping of those entries and you can phenotype some of them in those trials, you can develop prediction equations. And that's this quantitative phenotypic prediction, which enables you to predict the phenotype rather than have to measure it, which can make your whole breeding system far more efficient. You can do a lot more samples or measure a lot less, and so the expense to get genetic gain is reduced enormously, and so most major programs now use that whole genomic prediction as part of their system. What we're looking at doing is how to add biological knowledge into that system. The genomic prediction, if you look at the bottom there, the G2P with the whole genome prediction, is essentially going from one end to the other with a sort of bit of a black box in the middle. And so it's a bit limited in the sense that when you get genotype by environment interactions or trait by trait interactions, the sort of things that Charlie highlighted this morning, that system really has trouble in terms of its capacity to predict well. And so I suppose the argument here is we would like to introduce more biological understanding into that mix by bringing in combinations of crop models and trait network models. And that's what we'll talk about today. I'll cover the crop modelling part of this and how the crop models help with unravelling some of the G by E that can advance capacity for whole genome prediction. Nicole is going to talk about the branching network modelling that she's been doing and how that might connect in with the sorghum tillering example, for example, that I'll give here. And Owen will look at how this all synthesises into creating opportunities in advancing whole genome prediction. So for the crop growth modelling part of this, I like this picture from Corinne Chanou. Essentially, we've got algorithms that cover all the dynamics of the growth and development of the crop that are shown in that diagram. So you've got all those processes going on day to day: environment, radiation, water supply, nitrogen supply, all those things interacting to drive how much the crop grows, how much leaf area it produces, how much resource it uses in terms of water, nitrogen, light interception. All of those things have equations inside the crop growth model that enable you to predict the crop growth, biomass, and ultimately yield. And you can do that for different management combinations, different types, different levels of genetic traits, and different environments. One of the critical things, though, is this is not done in isolation from testing by validation with field data, and the example there just shows some predicted observed plots from a paper from Alex Wu. So once you've got these algorithms together and you sort of predict what happens every day through a crop, you can test that if you're on track or not and find out why not, and hopefully improve the capacity to model. I want to give a quick example on tillering in sorghum. So tillering is one of the traits that we've been looking at for some time. And the important part is what comes out of the end of this diagram here, the fertile tiller number, FTN, because that's actually what influences the size of the sorghum canopy the most. So it's not actually how many tillers are produced and how many are sustained, it's how many continue to grow and produce a rachis, and that will determine the canopy size. And that in turn interacts with water use, nitrogen use, the dynamic of that over time, and how you ultimately determine grain yield. And so there are three phases in here: the tiller production to get total tiller number, and the cessation of the outgrowth of those buds at some point, which can be due to plants realising that they've got competitors, and so things like red-far-red ratio will come into play to stop outgrowth of new buds. Once you've got a certain number, then there will be internal plant competition, mostly for sugars, so some of those buds that have outgrown will stop growing and you end up with a fertile tiller number. So that's essentially how we model it, and inside of that you get responses to genotype, environment and management. This is the sort of schematic of that, and I will have to use this. This picture from Eric Van Strom, really. You've got a sort of propensity to tiller, which is largely driven by hormonal control, which is the sort of network that Christine's group have been advancing, and I'm sure Nicole will cover that. But once you've also got a strong interaction with sugar status in this network, and so sugar strongly influences how this hormone network works, and sugar status is controlled by an awful lot of things. So it can be canopy size, leaf length, leaf width, environment, leaf appearance, and so all of these things are controlled by different sets of genes. And so you've got a large number of regions that control tillering. And then further out, you've got plant competition, which will, okay, this is propensity to tiller, but there will be some of those buds that at some point you'll stop, and then you'll have competition, so that some of the ones that have started to grow will cease. And in the end, you can do a simulation which shows if we have a normal tillering type, and each of these dots is a 1-year simulation, comparing that with a low tillering type and the yield difference. And the model just generates this G by E. So you've got, yield advantage from low tillering in these low yielding years below 4 tonnes, and a yield disadvantage at the other end. And so the point of the modelling here is that the model dynamics have inherent in it the processes that enable the generation of that G by E, whereas a whole genome prediction G2P model largely would miss it. So if we look at where we're bringing that modelling inside the whole genome prediction framework, we've got a model that can generate these sort of G by G and G by E interactions. So what we've done is really generate a G by E by M virtual database with those attributes. So we have a number of traits, leaf size, phenology, and we've then assigned a number of quantitative trait nucleotides to each of the coefficients that drive those traits in the model, and generated a 1,500 member population with some selection procedure that I hope Owen might explain to you, that enables us to generate combinations of all of those QTN across all of those individuals. So you've got a mixture of genetic architecture controlling all of those traits, and we can simulate it for a number of environments, a number of management systems, and a number of different levels of heritability, which enables us to introduce some error into the system, some noise. And so that's the database, the virtual database that we've generated using the model. And just to show that this actually generates the sort of variance components that one would normally see, this is the mixed model run that Mark did on that database, and you can see the variance components there. There's a large environment and environment by management interaction. And so that dominates the system, but there is also significant G by E by M variance components in here that relate to those trait by trait interactions. And I think this reflects back what Charlie was showing earlier too, that a number of these interactions are what generates the variances in what you see in these populations across environments. So where that's going is to bring that model inside the whole genome prediction algorithm, and this is the work that Owen is doing. And so the G to G by E by M, we connect that top line, the weights from those genomic regions to the coefficients in the model that enables us to simulate the trait by trait and trait by environment interactions to get the yield outcome of those interactions. Then you can cycle that around in a whole genome prediction algorithm to optimize, to try and find which of those regions are controlling those traits. So when we've set it up by assigning a set of regions that would generate the levels of the traits. But in the database, you get the phenotype of yield and the genotype. You're then in a position to look at different methodologies to say, well, which of those methodologies can best expose the regions that we know were actually set to generate that phenotypic variation. So essentially, we've sort of developed a model that with these sort of traits we can do that. I think there's room to move to improve that to more traits as we learn more and are able to improve the models. And we've developed this synthetic data set that enables us to test the different whole genome prediction methods. And there's really, that provides us with the potential to look at how you might go about improving this, enhancing the rate of genetic gain. And Owen will give you the answers as to how that's working. So thanks very much.
Have we got time for questions, David? one question. You're giving a great overview ready for the next two talks, so that's been wonderful as well.
All good.
I think we'll move on then. Thank you very much, Graeme. I think they're really wonderful outputs for the centre, so it's really great news.